So I think we're going to get started. I want to welcome everyone to the final panel of this fantastic conference day. So I'm Steve Dickman, CEO of CBT Advisors. My colleague Bruce Leuchter here is co-moderating. Bruce from Neurvati, Neurosciences, Inc. And we'll talk a little bit about ourselves, and then we're going to run through some questions that we think should be interesting about what's hot in neuroscience as we enter 2024. I want to start out by thanking the audience for coming and joining us at the latest possible moment in the day, and also thank the organizers, Leonard and Thomas and the rest of the team for making this, as usual, such an excellent event. So with that, I just wanted to say we're going to ask the panelists to say a couple of words about themselves and their organizations. We've assembled a group of investors and senior BD executives to talk about the market, and we're going to start at the far end with Tim. And please speak into the microphone. It's otherwise really hard to hear in here.
Yeah, hi, I'm Tim Leuchter from Lilly. First of all, thanks Leonard and the Sachs team for inviting us. So I lead the West Coast Ventures part of Lilly. So I'm sure most of you know Lilly's the world's biggest pharmaceutical company by market cap. We're in multiple therapeutic areas, but in the early stages, we invest in venture funds, we invest in individual biotech companies, we're strategic investors, multi-billion-dollar operation. And all of that is really to get introductions to great innovators, great companies that could be the future of biomedical research. Later on, hopefully, we can acquire or do BD partnerships with those companies. Me personally, I'm a scientist, pharma R&D trained. I've worked in big, medium pharma companies. I've spun out companies that were acquired. And really, I'm an early stage generalist and just excited about the prospects of helping patient health.
Thank you so much. Paolo?
Good evening, everyone. Thank you, the organizer, for the invitation. I'm Paolo Di Giorgio. I'm the CEO of Angelini Ventures. Angelini Ventures is a corporate VC, global corporate VC of Angelini Industries, that is an Italian-based mid-cap organization with 60% of revenues coming from a pharma division, with a strong presence in the neuroscience. In terms of my background, I'm a neuroscientist by training. I work at Merck, Novartis, and Angelini Pharma in R&D, and also with some BD experience. And I'm running now the fund since a couple of years. We are focusing in life science, biotech, but also digital health and medtech. We have a capital allocation of 300 million, and we invest globally. 70% of our portfolio is actually in North America, and more than 50% of our company are in the CNS space.
And Paolo, are you investing primarily early, mid, or late stage?
Yeah,
private?
So we have co-founded a few startup studios, 2 in Europe and now one recently in North America. So in our business model, we use the startup studio to access early stage innovation very early at academic, sometimes even pre-POC. Great. And the majority of our direct investment are in companies between Series A and Series C.
A to C. So, thank you. Thank you so much. Ivo?
Thank you.
Yes.
Good afternoon, everybody. My name is Ivo Staijen. I am not a neuroscientist. Maybe I should add that after 2 neuroscientists here. But I'm a biochemist. I work at HBM Partners, HBM Healthcare Investments. We are a VC firm out of Switzerland. We run about $2 billion, euros, Swiss francs. It's not so much different today anymore. And we invest in private and public companies. So I head up the public equity team, which is investing in mid to late stage public companies. And we also have a very prominent private equity arm, venture arm, that invests in early to mid stage companies. And of course, as an investor, our interest is always looking at new areas of development, and certainly neuroscience is prominent nowadays.
Thank you so much, Ivo. Doug?
Great. So Doug Crawford, I do several things. I'm run an incubator network here in the Bay Area called NBC Biolabs. And I also am a managing partner of a venture fund, Mission BioCapital, which has happily made many investments in the neurodegenerative space, despite the fact that on the road to raise, I think, Fund 4, one of my partners said, but we'll never invest in neurodegeneration. The clinical trials are too daunting for a middle-sized firm like ours. Happily, we didn't pay attention to that and have invested in a number of such companies, including Mitochondrion, which recently sold to AbbVie. And then I'm also the interim CEO for 3 years. Geez, interim is a long time. Of a company called Epidyne, which is developing a partial mu-opioid agonist for pain and opioid use disorders.
Great.
So I'm raising money too.
We'll come back to you on pain and also on your portfolio company Curalis.
Yes.
That's an interesting one in ALS. So we'd like to hear a little bit more about that one later on. Christina.
Thank you. Thanks for being here, Leonard, for inviting again. My name is Christina. I work as I would say, company creator mostly. I also have consulting roles with the Arch Venture Partners that you may have heard about, and as a Japanese American venture fund called Staijen Ventures. I'm very fortunate to be here with some of my investors in a couple of the companies I've founded. I co-founded Pretzel Therapeutics, which is a mitochondria focused company. And of course, mitochondria is important in degenerative diseases as well. I also co-founded Toon Therapeutics, which one of my investors is sitting right here next to me. So it's a good company here. Let's see what we can do with the other companies. Currently, I'm a CEO for another CNS company. It's called CNS2, very creative, focusing on glia biology. I'll tell a little bit more about that later on. So happy to be here. I think I maybe missed a couple of companies, but we can talk about them later.
Great. Thank you, Bruce.
Thanks, Steve, and thanks to Leonard and the team for having us again this year. Always enjoy this conversation. I'm Bruce Leuchter. I'm the CEO of Neurvati Neurosciences. It's a Blackstone-funded neuroscience operating company. We look across the neuroscience landscape, all different verticals within neuroscience for clinical stage molecules that hold promise for mid and late stage development. And then we commit capital for the entirety of the development program going forward. We have a first subsidiary company called Grin Therapeutics, developing a treatment for rare pediatric epilepsy called Grin-related disorders. And we're looking high and low for opportunities to create a second project around. I'm a neurologist and a psychiatrist by training. Spent a bunch of years on Wall Street, equity research roles and banking roles, M&A roles, and now for the last 2.5 years at Neurvati Neuroscience. It's always a pleasure to be with Steve as a co-moderator. I'll hand the keys over to him, and I'll do my best to contribute.
I promise to keep it moving. So thank you so much, all of you panelists. And I'm just going to give you a little overview of how we're going to do this. So we talked in advance about some questions that we were interested in. Given the limitations on time, I think we're going to wind up spending most of our time talking about what's hot in neuroscience. We'll give everybody a chance to tell something that they think is hot, and it's going to be hotter in 2024 than it even was in 2023. We'll talk a little bit about pain. Vertex got some pain data that seems like a long time ago, but it wasn't very long ago that Vertex's stock went up because of some great pain data, and we wanted to talk about the backdrop for that and the context for tackling a tough space like pain. And technologies. What are new enabling technologies that are driving more viable investment hypotheses in neuro? And so with that, I'd like to go back down the panel the other direction and ask Bruce to start telling us about something he thinks is hotter in 24 than it was in 23.
Hotter in 24 than it was in 23. That's I'll tell you what's hot. I think large indication opportunities are hot. And that's not a scientific or therapeutic modality driven comment. It's more of a comment on where investors are deploying their capital, where strategics are transacting. And you don't have to look much further than the last quarter of 2023 to validate that statement. I think Vertex has been mentioned a couple of times already on the pain data, and I think it was a $13 billion uptick in market cap on a phase 2B data point. We saw the transactions at the end of the year targeting schizophrenia, a big indication despite all the generic presence in the market, lots of appetite for novel mechanisms. So I think it's less about whether it's a large molecule, small molecule, genetic medicine, et cetera. And in my view, at least, it's more about what moves the needle. That may feel like a less romantic approach to how we think about drug development in neuroscience, but I think sometimes it's okay to park romance to the side and think about how things actually get through the system. And when I think about it in that way or view the world through that prism, I look at large indications, I look at late stage studies that can prove the thesis out, deliver positive data, and that gets people excited, full stop. So you can see companies that are aiming to find their way there, neuropsychiatry being a great example of that, given all the unmet need. And if I was forced to think about something that was, technology specific or clinical trial design oriented, I'd come back to what I think I said last year and probably the year before. Any attempt that we can make and succeed in delivering on an enrichment strategy for our patients, the better off we're going to be. And you'll see more of that, I think, in 2024, how to refine patient populations, how to understand parsing of patient populations, and driving more effect size in mid and late stage studies. Because I think our space continues to suffer, perhaps more so than any other space, from failed studies, not failed drugs. But the bottom line, as I see it, is there's a ton of unmet medical need in neurology and psychiatry. If you can position programs against that unmet medical need, you're going to have a lot of enthusiasm from all the stakeholders.
Okay, so that sounds hot to me, and I invite any panelist to bring the romance back. Anybody wants to bring romance, I'm good with it. You don't have to, it's optional, but desirable. Christina?
Yeah, so from my perspective, I've been focusing a lot on other cell types necessarily than the neuronal population. So obviously I have with my connection, I'm also adjunct assistant professor at Center for Translational Neuromedicine in Rochester, and we focus a lot on understanding glia cell biology, not the microglia part at this point, but more the macroglia. What is the function of the supporting cells in the brain? I think they have been understudied in both their importance in, for example, schizophrenia, other disorders like that. In particularly, the glia progenitor cell pool is also something that we are looking at. We know when we age, when we reach and you see it from a lot of MRI studies, when you reach 50, 60 year old, you start to have a lot of white matter loss. So what is that, and how can we sort of, you can say, circumvent that? And that could be, for example, by using some of the hot terms, speaking of cell rejuvenation. So focusing on elucidating cell pathways, mechanisms that can potentially remobilize cells in the brain that is aged, I think is something that we'll see more of as well. But it's not enough to understand all the networks and the cell contribution to pathology. I think another part that's critically important in this space is also the delivery aspect, which has been sort of pointed towards in some of the earlier sessions here. And we had it, we saw Voyager and now S.p.A also had some news in those spaces. So I think that's an area, at least where I've been focused on developing new human-specific capsids for other cell types than just neurons, and looking at, for example, delivery pathways that are not necessarily IV, which of course is the holy grail, but maybe looking at, for example, cisterna magnet delivery, whole brain delivery, and targeted cell types. The other aspect I think is really sort of emerging, which taps a little bit into the glia cell biology, but almost has its separate space, is understanding what is the waste mechanism place in the brain. So, for example, looking at the lymphatic clearance. So this is one of the, I think, emerging areas that can actually have a broad impact if we understand how to increase the efflux and that waste clearing in the brain. So that could be for TBI, Alzheimer's, all the proteinopathies, etc. So these are some of the areas that I pay attention to at this point.
That all sounds like it qualifies as super hot. I would also thank you for returning our attention to Voyager and AskBio, companies that have been around for years. AskBio was actually acquired, and at the same time, there's a really great, that's a former client of mine, there's a really great gene therapy discovery engine inside that company, and Jude Samolsky, who more or less invented gene therapy. It's a really phenomenal effort, and it's just fun to think about how it disappeared a little bit from the public eye after being swallowed up by Bayer, but now here we are with some very exciting news. Yeah. Doug?
I think the big innovation we're going to see in 24 that we didn't see in 23 will be optimism. Man, was this 23 a year to forget. And But I would say on the other side, the other thing that will drive optimism won't be just the restoration of the S.p.A, but the existence of success stories. So Lilly's great data is really, raising the boat. I mean, for many years, my partner's view was the standard view, which is that this space is just too hard and expensive to be in. But now we've demonstrated that you can provide a therapeutic to a patient that restores the most complicated machine in the universe. That's another reason for optimism. So that's going to be my vote.
Super excellent. Thank you. Ivo?
Yeah, I think and I hope that the 2024 is just going to be a continuation of 2023.
Oh no!
If I may say so, but from a different perspective, I guess from a later stage investor, we've had a great year in neuroscience, and I think this will continue. There's more and more attention to neuroscience on various levels. We've seen the first products for Alzheimer's been approved. These antibodies. It's not a miracle cure. We need to go further there. Absolutely. The muscarinics are back in vogue with basically two acquisitions, maybe more to come. So from an investor point of view, I think it continues to be a great setup, and yeah. So I hope it will continue. Sorry.
Thank you. Paolo?
Yeah, I mean, there are probably multiple answers to the question. I agree on the late stage. I mean, we're seeing December like a big M&A acquisition of Karuna Cerevel, and definitely it seems to be a great moment from neuroscience. I think in general, also from the early stage perspective. I mean, of course, the field is continuing to deliver and to create new first-in-class, best-in-class with standard mechanism. But I mean, an area where there is a big need is, in my opinion, to what is a buzzword, but it's quite important, particularly in the neuroscience field, is personalized medicine, and try better to understand patient stratification and how to deliver the right drug for the right patient. This can happen in two different ways. One is on like the clinical symptomatology and also biomarker development, and the other is more on be able to really modulate the neurocircuit in more specific ways. So these are the two areas that I'm excited about. I don't know if we will see many changes in 2024. It's more like a long-term perspective, but I think it's going to be fundamental in the development of neuroscience. At some point we will get there. We see already some companies moving in that direction.
Great. Tim?
I'll give you a perfect storm of optimism. So I think we've been working for a number of years on different kinds of payloads, as Christina alluded to. We can do DNA editing, we can change a single base, we can insert a whole chunk of DNA, we can almost replace almost entire genes. And then at the RNA level, you can do transplicing, you can do knockdown, you can do upregulating, you can do editing. And those technologies are all coming into vogue right now, and they're better understood, and they're starting to show benefits in diseases where it's easier to deliver to the organ of choice, like the liver. What's also moving at a really incredible speed, as again was alluded to, is the drug delivery aspect. So, there's ways of conjugating things on to help delivery. Everyone knows transferrin and some of the early data, but there's a whole raft of new targets that are coming forward to get across the blood-brain barrier. Capsids like the Voyager, but there's also a load of new different viral vectors that are coming forward that may be neurotrophic, they may have different properties, larger cargoes. And then even the old lipid nanoparticles that everyone looks at the ceiling going, God, that's 1920s technology. Those are starting to look really good now. And not only lipids, but polymers and particles that compress themselves and get in spaces, exosomes. It's hard to say which one of those is going to be the absolute winner. But there's a perfect storm of progress that some of those combinations will be put together. I mean, to put a gene therapy into the brain, you need a vector to get it there. You need a good genetic target. You need the right way of modulating it. And now we've got things like promoters and enhancers that stop some of the other organs expressing the transgene. So even if a bit of that goes to the liver, nothing happens. And you can tell it, hey, only express your payload in the diseased neuron. When all that stuff comes together, I think the brain's going to turn into the liver in terms of a sort of area of optimism. And I'm pretty confident that, as I say, we're not quite there in saying exactly what the winner is and how that's all going to fit together, but I'm very optimistic that a proportion of that is going to come together to disease-modifying treatments that we're going to be, maybe even some of us will start taking in our lifetimes.
Yeah, I think that's an important point. Just reflecting on some of the comments down the line, it isn't just about a particular therapeutic modality or, for that matter, even an indication. I think it's about momentum and building upon prior accomplishments and successes. And that momentum can be contagious. I think we did see that in the fourth quarter, and I think we're going to continue to see it in 24. And a lot of it is built upon the successes and, frankly, sometimes the failures of companies that preceded us on this journey. Whether it's gene therapy and figuring out how to target brain more effectively, or whether it's novel mechanisms in neuropsychiatry. Just the notion that there's appetite, the notion that Bristol, God bless Bristol, who's been, I think their last exposure to neuroscience proper, if you remove the S1P for MS, was Abilify, neuropsychiatry success, a tremendous success. But that's many years ago, and now there's been this desire to wade back in because there's so much unmet need and there's so much enthusiasm. And if you look back on 23, not just the fourth quarter, I think you'd see it across different kinds of indications. So I agree. I think that we're moving in a direction of not just incremental progress, but really finding our way toward enthusiasm that should support the whole therapeutic area.
Thank you so much, all. And before we start to dig deeper into pain and into enabling technologies, I did want to relay a little anecdote, which is also a way of introducing my firm, because I didn't do it before. In 2019, my firm was involved in drafting the business section for the IPO prospectus of Karuna. And I just want to point out that our efforts began in about April of 2019, or in February, I suppose, and then they filed in April, and then they went public in June, and the stock was flat. The stock was flat for about 6 months. In fact, the stock traded slightly down until the phase 2 data. So I just want to call attention to that at a time when there was no momentum for a novel neuropsych mechanism, and especially pessimism toward muscarinics, that stock traded down until, lo and behold, the combination of xanomeline and trospium chloride turned out to be a winning combination for schizophrenia patients and then eventually other potential patient populations. And the stock went in one day from $15 to $85 a share. That was in November of 2019. So I remember that day well because I really believed in the company, and I thought the story as a story was one of the more compelling stories that I had the pleasure of working on. And I just again want to underline Bruce's point that is in some way illustrative of how much more momentum there is in the market right now, that company less than 5 years later gets acquired for $14 billion. Now that's momentum, but obviously driven by very strong clinical data in an indication, as you said, where there's huge unmet medical need and a mechanism that has a lot of logic to it. The mechanism makes sense in a way that I don't think people believed in 2019, and now the market is much wider open for mechanisms that make sense. And that's really good for everybody in the room, everybody in the industry, I just want to say. So I did want to come back to Vertex and ask and now I'm not going to ask every panelist to comment, but whoever wants to comment on the sort of larger meaning of this. We know that Vertex was doubted. We know that pain is a very tough indication. We know that the industry in general has been averse to even developing pain therapeutics because there have been so many hurdles and hassles and addiction issues and other issues and side effects. And Vertex plunged ahead and they put a lot of effort and a lot of money into their trials. And they got, again, the increase in market cap that day was a reflection of how pessimistic the market was on how well that product would work, VX, 548. So I just wanted to ask the panel, so what does it mean? There was a pain readout and Vertex's stock went up, that's great. But let's dig a little deeper. What does it tell us? Where are, as you were saying, Bruce, the thresholds for safety and efficacy? Have they moved? Has the bar moved because Vertex got good data, or had something shifted before? Help us understand what's going on. Bruce, maybe you want to start.
Yeah, I come back to my opening comment. It's not romantic. The data worked. The market opportunity is incredibly large, even with generically available pain medicines in the space, and the opportunity is huge. It doesn't take much more than that. I'm not sure actually that it's a lack of interest in novel mechanisms or It's a lack of intellectual curiosity. I think it's driven by the opportunity to reach patients, treat patients, and derive economic upside from that. And that again, a little bit of provocative posture from me at 4:30 on a Sunday. But I think the good news is that it actually isn't always about the totality of the program that you're contemplating. It's about whether or not the next data point that you can deliver can be sufficiently seductive to the stakeholders that you're targeting to drive the level of motivation and enthusiasm that can further support your company financially or otherwise. And I think you see that. You clearly have seen it in neuropsychiatry. I think you've seen it now in the pain space, pursuant to your question. But pain is hard. It's not any. I think it's comparable to schizophrenia in that way. Very, very tough to keep the effect sizes as high as they are in phase 2 studies. As you move into phase 3, it's challenging to reproduce those data. The existing therapeutic options in the space are multiple, and at one point were themselves multi-billion-dollar therapeutics. And it does raise the bar for the kind of efficacy that one needs to demonstrate in order to generate the kind of animal spirits that I've been referring to. Not the romance, but the animal spirits. And when you can do that, you can drive the necessary level of enthusiasm to actually accomplish what everyone in this room is keen on doing, which is developing successfully novel therapeutics to treat patients. So I guess I sound a little bit like a broken record, but what I'm trying to communicate to those folks in the room is that it isn't always about the totality of what happens between time point 0 and the time your drug becomes a multi-billion-dollar opportunity. It's actually what happens incrementally. And if that incremental improvement, incremental success can yield the kind of financial support and other sorts of resources that can allow for those sitting in the room to succeed, that's a very good thing, even if it lacks some of the sex appeal that a very early stage high science platform technology might provide. It's very good news for everyone here that because there's so much unmet need and because there's so much desire to step in after a data point that proves thesis out, proof of concept data point, we should all be focusing on how to deliver that data point. And my belief, and I think it's been proven out, is that if you build it, they will come. And that's exactly what's happened in the Vertex space. You know, everyone and their kid brother had basically given up on sodium channel modulators for pain. It was, not viewed to be a place where you would deploy capital. And yet the stick-to-itiveness of Vertex and ultimately the data, have clearly demonstrated how much pent-up enthusiasm there is. And I think that's true across disorders, no matter whether we're talking about neuropsychiatry, pain, or for that matter, genetically defined neurology.
Okay, we'll circle back to what you could do to get to that. I would hesitate to call it this because we can't say it in an S1, but de-risking event, which is, yes, proving your proof of concept, but also removing some of the challenges for future clinical research. But I want to ask the other panelists before we move away from Vertex and its amazing pain data, is there more to be said about what happened, about why the industry now is positive about something that it seemed like it was very negative about for a very long time?
Well, we shouldn't forget the pain and suffering that industry faced with NaV 1.7. Yeah. Genentech, Merck, Amgen, lots of dollars spent, nothing to show for it. How do I know that? We're an investor in a NaV 1718 company, Site One, so I followed this story pretty closely for many years now. Unfortunately, we're riding on Vertex's coattails to the promised land, let's hope. But Vertex deserves a lot of credit because if you look at that sad story for NaV 1.7, it would have been easy to give up, and they persevered. And very few others did, so they're years ahead in this battle. I think for patients, it's going to offer more opportunities, but more nuance as well. So the industry is driven to these hyperactive opioids in a kind of, there's a weakness between both the patients and the physicians. The physicians don't want to get calls in the middle of the night, and the patients like the euphoria. And so it's like, okay. So Vertex is not claiming that they're going to displace acute pain treatments. We're going to have opioids forever. So what is that dividing line, and how will physicians deal with phone calls, which they're not getting right now? It's going to be really interesting to see.
Yeah, that's exactly the kind of parsing, Bruce and Doug, that I was looking for, is to try to understand what's going on behind that headline. Anyone else, or should we move on to enabling technologies? Tim?
I think we're mixing up quite a few different concepts here. So, on the xanomeline story, for example,
Yes.
that's a target from the 1980s.
Yes.
It's like, this isn't new thinking. It's more...
Yeah, that was a Lilly drug
Yeah.
that was shelved because of terrible GI side effects.
GI, heart, salivation, that was a sort of controlling the mechanism understanding story. I think the NAV story, it's always been a genetically validated target. How safe it is, time will tell. But that's a problem in drug discovery. Like small molecule ion channel blockers, it's a high attrition area. Selective, there's so many subtypes of these, there's so many binding sites. You have pore blockers, you have allosteric sites. It's a very complicated area of drug discovery. We'll see if Vertex have genuinely threaded the needle. And there are other targets, the CAVs, the NaV1.8s. There's a whole family of these that are good targets if you can actually drug them. And I think our small molecule drug discovery de-risking has improved a little bit over the years, but still a lot of those molecules fall out for molecular tox and then selectivity issues. And I think, they're always challenging, but there are some people that think any target takes 10 years to get a drug out the other end. I don't think these are any better or worse than some other areas that we could talk about. And then in terms of de-risking, I can say a little bit about what Lilly's doing in pain. So we're still committed to pain. We actually have a sort of clinical trial master protocol where we put novel mechanisms through 3 or 4 different pain indications with very good understanding of the stats required to get a signal of a significant size to warrant the next stage of development, because it isn't always clear which type of pain novel mechanisms will treat the best, actually. So there's a whole load of things to unwrap in those discussions, but certainly I think the NAV story has really been a drug discovery development kind of molecule story, really. It's always been a decent target overall.
Well, I would have to say your description of this master protocol, it fully validates Bruce's point about, it really embodies the point about the efforts required, the actual work required, not romance, to push through all of these compounds, to have a huge database and a huge vivarium, honestly, and then really be in a position from expertise earned over years of effort to essentially analyze accurately what's going on and make the right choices about the next steps for drug discovery.
And also that there's not a shortage of pain targets, actually. There's a shortage of human data on pain targets. So obviously there are families with rare mutations in some of these targets that tell you that they don't sense pain, so you kind of know it's going to work. That's not true of a lot of the other targets. So I think bringing forward more human-centric understanding is critical, really. If I talk to our pain people, they're like, we've got zillions of targets we kind of like that look okay preclinically. We have no idea what they might do in patients. So it's usual drug discovery stuff.
Yeah, and you make a valid point, Tim, about the ancient nature of the xanomeline program at Lilly and the lag time to achieve clinical success. I would say that next we should consider the early stages of drug discovery and the technology-driven approaches in neuro. Specifically, I started in our panel prep call talk about cell and gene therapy, and Bruce accurately corrected me and said, no, Steve, there are lots of technologies that are brought to bear on understanding of how the brain and CNS work, understanding of how to alter the biology. You want to talk a little bit about that? And then Christina, I want to hear from you too. You're actually active in this space.
I'm going to pass the baton to Christina, and she can take it. She can take it from there.
Please, what are the technologies that we should be looking at? Biomarkers, anything that's going to contribute to the de-risking and speeding up of these traditionally very slow drug discovery processes.
Yes, I would definitely.
Microphone.
Definitely the biomarkers is a big area, and even when you consider the hot field of health span and these aspects, that is still very new for us to look into. But obviously, a lot of the technologies of multi-omics has really enabled us to look more deeply into not only the cell and the molecular pathways and the chromatin, but also the structure of the brain as such. And I think this will, with time, increase our understanding on how this incredible machine is working. At the same time, I think a little bit to Tim's point, over the past years, there's been a tremendous effort on understanding RNA editing, gene editing, epigenome editing, and all these different technologies based on the biology understanding that we have. So I think one of the things that's really starting to missing and we're now directing our focus on, and we saw that the last couple of years, is the one company after the other promising new and more targeted delivery mechanisms to the brain. So I think these are still some gaps are over there, but I know several groups are working very dedicated to solve some of those issues. So I'm extremely excited for all of it, and I think looking more holistically too, but broadly on other different cell types and how they interact is going to be a critical driver for see some significant progress. Not only the neuron is important.
That's awesome. Biomarkers, anyone want to tackle that? What we expect to see in terms of improved biomarker discovery pointers that can accelerate and improve quality of drug discovery? It's another hot space.
Well, maybe just to add on technology and then to converge on biomarker. I mean, to add to the point of Christina, there are many exciting technology coming out. For example, a combination of gene therapy with optogenetic, chemogenetics that could use for research purpose, but in more advanced setting could even consider as a therapeutics. And again, all the combination that allow us through omics and also to analysis of clinical setting to understand better patient, to better resolve patient stratification. So these are all aspects that I think could be enabling new type of therapeutics.
Okay, optogenetics is still very cool.
It's cool
And
it's
romantic.
but you know, if we want to understand well the circuit, could be an amazing tool. If we want to use it more for a kind of specific target, therapeutic setting, then it's much more complicated. But I think one of the opportunity that we still have with the brain is really to understand and to modulate different neuronal circuits. And we know so much now based on what has been the neurobiology in the last 60 years. If we are able to modulate circuit to technology like chemogenetic or like protein degradation specific for some subtype of neurons, you know, there could be a new wave of neuroscience.
Thank you, Paolo. Before we get to the biomarker question, I gotta ask this because I've been following closely the breakthroughs in artificial intelligence research and looking for areas where AI can actually productively near-term impact drug discovery. And to be honest, it's hard necessarily to just find those areas. Like, you got to do some digging. But as you talk about this, it occurs to me that the original neural networks, the original sort of adaptive learning models that preceded the LLMs that have taken over our world today, they actually emerged from study of the brain. And the more perceptive neuroscientists were pursuing an ability to learn and adapt in the perceptrons, for example, in settings which were artificial and yet mimicking what happens as neuronal networks are adapting and learning. And so I gotta ask, isn't this an ideal application if you're trying to mimic the behavior of neurons in a non-biological setting, in an in silico setting, for example, and see how they learn? Couldn't you also perturb the system and see how you could perturb the system? And couldn't that lead to better drug discovery? I'm fumbling here a bit, but it just seems to me like there's a logical connection.
Well, I mean, in theory, you can use that to model neurocircuit. That could be one aspect, as you mentioned, and try to replicate. I mean, if you have enough computational power really to replicate neuronal circuit. Of course, now AI has developed in multiple, as we know. I mean, it became, it's almost a buzzword. But I think if we think at more generic AI, the application that can have in drug discovery, we're starting now. Probably we'll see much more advancement in the next few years.
It's starting now. That's a good way to look at it. I don't want to abandon biomarkers because I think biomarkers are the future. I think that they're driving some of the more exciting public neuroscience companies, the more exciting early stage companies. Last year on this panel, I mentioned Vigil, and I'm still watching Vigil because they're a biomarker-driven drug discovery company trying very hard to come up with exactly those kinds of events that Bruce was talking about and show the kinds of impacts on tough targets like TREM2. We've got a lot of tough sledding ahead, and at the same time, the biomarker area seems to have, EEGs and patient-specific biomarkers and the biomarkers that are potentially digital in nature. And I wonder if anybody wants to speak to that about where they see potential advances coming in the near future, maybe not just in 2024, but in the future.
I think I go back to the failed studies versus failed drugs, and that's the bogey that I think we all struggle with in phase 1b, phase 2a studies, where there's a lot riding on the data that gets delivered from those trials, a lot riding on it. And patients are counting on us to deliver data that allow for further resource allocation. Stakeholders are counting on us for the same. So how do you manage a heterogeneous patient population phenotypically as they look clinically? How do you solve for that? Because what's happening inside endophenotypically is going to be different across those patients. And if you're able to solve for some of the endophenotypic heterogeneity by way of homogenizing those populations through things like EEG and ERP, it isn't the whole solution. I am not sitting here telling you that it is a magic bullet. What I am saying is that it's a triangulation exercise, and the idea that we can simply pick a diagnosis, create or identify rather a very, very compelling target, very highly scientifically informed, and pursue it in a patient population with a certain diagnosis is naive in my view, and is oftentimes what leads to underperformance and therefore lack of resource allocation to these very important programs. So if you think about something like EEG and ERP, it's a component part of the solution, because at a minimum, you can look at the endophenotypic readout from those patients and you can do something with it. You can do something to inform the way in which you're thinking about enrolling patients in the next study. You can try to enrich patient populations. We're already stipulating that the innovation in our world, in our space, is off the charts. It is absolutely romantic, and it is absolutely seductive. What we're having a more difficult time with is translating that into the kind of effect sizes in early stage studies that drive all of this fervor, all of this enthusiasm that I think our space is experiencing now more so than it has in prior years, and I think we'll continue to experience in 24. But the more that we can triangulate, whether it's with imperfect but nonetheless existing plasma or CSF biomarkers, imperfect but nonetheless present EEG and ERP technologies, and imperfect but nonetheless available neurodiagnostics like neuroimaging, the more able we are to parse those patient populations and realize success. We're going to hear data. We've heard data in 2023 from Alto Neurosciences on this exact topic, where they've taken medicines that have not worked in the hands of very well-funded companies that could not deliver a positive data point with some of the most seductive biology you can possibly imagine, but no proof in the pudding. And so those programs were shelved and people were disappointed and patients weren't helped. But what Alto has figured out how to do now in 3 studies, 3 de facto proof of concept studies, is use proprietary technology that actually has nothing to do with the mechanistic rationale and the biological rationale, which we all agree makes sense for these indications. It has everything to do with the patients you're choosing to study to determine whether or not it works. And if you can deliver the effect size, to Tim's point on what xanomeline being 20 plus years old, if you can deliver the effect size, which Karuna was fortunately able to do with blocking M2, M3 agonism, then the effect size is what drives the interest, right? And I'd argue that these enabling technologies, biomarker technologies, whatever they are, none of them are perfect. But if we allow the perfect to be the enemy of the good, we're going to find ourselves with more in the way of failed studies, not failed drugs than we'd like, and we're not going to realize the success that we want. So I'm a huge proponent of thinking holistically and well beyond the seductive biology and well into the trial execution that's going to deliver the kind of data point that can continue to resource our companies.
Perfect. Comments? Tim?
I'll start by saying I couldn't have put it better myself. Great summary. I mean, there's obviously 2 kinds of biomarkers. One is a bit better understood, like target engagement biomarkers. I mean, we still see companies coming forward for investment that can't prove that their molecule interrogate the target. That may need CSF. It may eventually turn into plasma. But And then there's sort of enrollment biomarkers for clinical trials. It wasn't that long ago in the first Alzheimer's trials that people were being enrolled that had some sort of cognitive impairment. There's probably like 7 different diseases under that, so it's not surprising the trials didn't work. I mean, Lilly's obviously brought forward like amyloid and tau tracers to actually have enrollment criteria, so you actually test the hypothesis correctly. There's a whole patient access story that's a problem there, because who's going to be able to get those, and hopefully a plasma biomarker will straighten that out longer term, but we'll see. But stratifying patients just through clinical phenotypes is not great. They did the best they could do at the time, but it's not surprising that an anti-amyloid antibody didn't work in depressed patients, for example. So that's the first thing. And then the relevance of the biomarker to disease, that's a much more tricky story. So I'm glad you mentioned Alto. We invested in that company. They obviously use AI. They've got a very complicated sort of EEG kind of way of stratifying subpopulations of, I think, schizophrenics in a clever way. We'll see. I mean, it seems to be working, but again, the mechanisms they're working on are pretty old and well-known mechanisms. We'll see how that goes forward, but it is a very tough area. I mean, you can't take people's brains out and see what's going on. So it's definitely a challenge. I think we've made some good progress from the old days of just anyone who looks a bit sick gets into a trial, but I think there's obviously a lot more science to come, I would say.
Awesome.
I would like to add to that. There's nothing that turns off an investor as a failed trial. I mean, that can really kill your concept, even though the concept itself is valid. If you pick the wrong patient population, even the wrong subpopulation, the wrong endpoint, the wrong duration, people were thrown away and think, okay, so that didn't work either, and they move on to the next thing. Unfortunately, very often, a thing you often see, and as you can see by a number of drugs that have been resurrected the last couple of years, that there is value in many of these concepts. And also I have to plead a little bit for my own job as an analyst, be that a sell side or a buy side analyst. Analysts also have to be re-educated in the neuro field. Maybe not so much in Alzheimer's, because we've had to deal with these epic studies of Biogen and whatever happened there, and ESI, but in other fields of neurology, and I include myself in this. I mean, it's not to the forefront of many public equity investors.
Yeah,
it's been more oncology and immunology and gene therapy, and the crash course in obesity last year, and But CNS is, we need to get our heads around it also from the investment side.
Unfortunately, the Alzheimer's data that's been delivered to the biotech buy-side and sell-side analysts generally has caused an immune response. So only recently has there been data that actually makes them want to buy the stock. So that you got to admit that it's, education is not all, but you're certainly right that there is room for the kind of work that several of us up here do in explaining and putting into context the nature of previous failures, in fact, renaming them from failed drugs to failed studies, and then moving forward with smarter studies and more provable hypotheses. We're coming up on time soon, so I did want to turn to the audience and see if somebody had a question, and also turn to the panelists and see if somebody had another comment before we open up any other big topics. Looking for any hands? Okay. And from the panel, I think before we dive into something new, I think we should settle where we are. I think we had a great year for neuro in 23, way better than any of us would have predicted a year ago. And especially with the acquisitions, but also with some of the data readouts and early stage financings leading to really exciting hypotheses that could get proved in the clinic. Anybody wants to throw in some really exciting young company that they want to talk about? I know Christina talked about 3 or 4 already. We don't want to let her monopolize it, but Curalis, tell us a little bit about Curalis. Why are you going to be successful in ALS with that company when so much else has failed?
Well, this is a lovely partnership with Lilly.
Microphone.
It's been a lovely partnership with Lilly, and we're super sanguine. I think one of the things that we haven't mentioned yet is neuroscience has gone from a phenomenological, empirical science to one of target-driven hypotheses, where we could argue that we're on the doorstep of a kind of precision medicine that oncology now takes for granted. And I think Curalis is a good example of that. And ALS is, God, it's an indication that really needs our attention, and we're happy to be part of that.
Great. Anyone else? Exciting early stage efforts? Christina, tell us a little bit about one of yours. Pick one.
CNS2. Tell us more about CNS2.
I think I basically told most about the CNS2. It's a glia-centric focused company. It has sort of two different areas of focus, which is the lymphatic system, and which is really closely linked to the biology of the astrocytes. I think that's going to be a big thing. The other aspect of CNS2 is really understanding pathology caused by astrocytic dysfunction and also by aging of the, you can say, progenitor population in the brain, which is the glia progenitors that gives rise to oligodendrocytes and astrocytes. And these are really critical support cells on the brain that takes care of our neurons. And oftentimes they fall off and they don't behave well. And then we get schizophrenia, we get degeneration, all these kinds of different diseases. So that's really the glia focus that I wanted to bring to attention. I'm working with a separate company out of Stanford, which is related to mitochondria fission and fusion, which is also something to pay attention to in some of aging and in neurodegenerative diseases. It's a different approach than what Pretzel has taken, but in my mind, being very committed and dedicated to try to build CNS companies, I think the more shots we have on goal and give them a chance to mature out of academia, the better it is for all of us.
Couldn't agree more. Well, with that, I want to thank the audience for your attention. I want to thank the panelists very much for participation. Thank you for co-moderation, and turn it back over to Leonard for drinks. Upstairs.